In this module, we will study that what are the parameters and what are the different factors which, as a matter of fact, affect or put a ban on the number of time a cell can divide. That is the limitation on the number of cells which can divide during the process. Now you see that most of the human cells, they divide a limited number of time before they stop and undergo permanent cell cycle arrest. It is naturally true that all the cells which have been produced or which are there in humans or animals or other, they have a limitation. They cannot grow for all the time or for indefinite period. So therefore, there is a permanent cell cycle arrest. That is, a stage comes when those cells, they do not grow anymore. For example, fibroblasts from normal human tissue go through only 20 to 50 populations doubling when cultured in a mitogenic media. That the medium when outside the human body or outside in a flask or outside in vitro in a mitogenic media, which are enzymes or proteins, they can go only to 25 to 50% or 50 populations. 50 times they will grow and they will double. Now, replicative cell senescence, that is, the senescence is a process where a loss of cell's power of division and growth occurs. Now, a limitation in the number of times that cell can divide appears to be the basic feature of somatic cells, except for most tumor cells and possibly some stem cells. Because the tumor cells, at this point, I may add, they grow forever, they are lifeline. Whereas the stem cells, they have also the capability to grow again and again, a limitless growth. Now, replicative cell senescence in human cells seems to be caused by factors like change in the structure of the telomeres, their structure changes, the repetitive DNA sequences, because DNA is a major player, and associated proteins at the ends of the chromosomes. They are the major factor which are responsible for that. Let's see that here we have shown that in the telomere, as they go to the division, again and again, the proteins at the end of the telomere, it becomes shortened, and eventually the gene sequence, it changes and the process, it stops. The exposed chromosome ends are sensed as DNA damage. So because as those proteins, they are removed, or it's shortened, so there is a DNA damage which activates again a p53 dependent cell cycle arrest, and the cell cycle stops. In contrast, rodent cells maintain telomerase activity when they proliferate in culture and do not have telomere dependent mechanism for limiting proliferation. It means that the telomere dependent mechanism is also an important factor in limiting the proliferation or the growth of these cells. Just as shown that in this picture, we have seen that there are many factors which have been combined together, that the cell proliferation, DNA damage, oncogene, strong mitogenes, they all lead to potentially cancer-causing cells. Then, on the other hand, the normal cells, which are mortals, which die, they undergo the three processes. One is the senescence and loss of that. Then is the apoptosis. Apoptosis is a programmed death of the normal cells, whereas the potentially cancer-causing cells, which are immortal cells. Mind you, the cancer cells, they are immortal. They grow forever, and they also undergo the transformation or the apoptosis. These are the three stages where the tumor suppressor mechanisms occur or lead to. Now, cellular senescence is an important tumor suppressor mechanism induced by potentially oncogenic event. And we say that the many oncogenic genes, they act by allowing cells to bypass the senescence response. So it means that the cells that do not have senescence response, they can bypass this cancer activity, and the senescence is controlled by the two most important tumor suppressor genes. One is P53 and then PRB. Now, it has been noticed that the mice with cells that do not senescence, they die young of cancer because this activity, it becomes evident in them.